Okay, so this is my brief rundown of how mitosis works. So what you're looking at is the whole white mass is your cell, which would be enclosed by a cell membrane, and it'd have obviously cytoplasm and organelles and everything in it. And these are our 3 chromosomes, which would be encompassed in the nuclear membrane, so inside the nucleus. So basically I have 3. An adult developed cell has 46 chromosomes. I only have 3, but just know that in an adult cell there would be 46, except for sex cells, which have half. But so in interphase, the cell starts to grow, kind of enlarge, and more cytoplasm and more organelles are created so that whenever the cells separate, there's enough for both cells. So then in late interphase, the chromosomes will duplicate, and so the orange will have an orange pair, and the green will get a green and create a pair, and then the yellow will get another yellow and will create a pair. And these are called homologous pairs. Basically, they are pairs of 2 chromosomes that are for the same thing. So like both of these would be for eye color or for hair color or for something of that nature. And though they might be different alleles and have different codings down the chromosome, they are generally for the same thing. So these sister chromatids that have come together are going to eventually, okay, so we're going to move into prophase. And in prophase, they're going to coil up. Like in interphase, the first phase, they are arranged as spaghetti noodles in a bowl. Not very organized, just kind of all over the place within the nucleus. So as we move into the second phase, prophase, the nuclear membrane starts to disintegrate, and the chromosomes, the chromosome pairs, start to coil up and get that H or X or cross configuration that we're used to seeing chromosomes looking like. So that's what's happening in prophase. And then these spindle fibers, these protein spindle fibers out here, these little fingers, are starting to form. So then as we move into prometaphase, the chromosomes have already condensed, shortened, and they're very coiled, they're very formatted. They're the form that they're going to be throughout the rest of the process. And the spindle fibers are going to come in, and what they're going to do is they're going to grab these pairs at the centromere, which is where they cross. It's the chiasm. And by pulling and tugging, we're going to move into metaphase, which is when they align the chromosome pairs along the midline by kind of like pulling an equal give and take. It's going to create this line. And then as we move into anaphase, these are going to get tugged apart. Now, I just want to make one thing clear. When I tug them apart, I'm going to be taking them apart like this. However, that's not the case in real life. In real life, that would be one chromosome, and that would be the other chromosome. So they would be splitting as if we were cutting it from the top space to the bottom space, rather than like this, just to be clear. But so we're in anaphase, and the spindle fibers, which are these fingers that have come out and have grabbed these at the centromere, are just going to grab, and they're going to shorten and tug one of each pair to the opposite sides of the cell. So then we're going to move into telophase. And telophase is when at each pole of the cell we have identical sets of the sister chromatids. And the spindle fibers are going to disappear, disintegrate and kind of not function anymore. And the nuclear membranes are going to start to form around these separate, which will be new nuclei. And then as cytokinesis happens, the cells are going to start to move apart. And we're going to get this formation where they start to pinch together. And because we have the sister chromatids at different ends, as they separate and pinch apart, we'll have two completely separate cells with identical sets of chromosomes. So as the nuclear membrane forms around these and these pinch off, we enter into two brand new interphases. So we've got interphase over here of cell 1 and interphase of cell 2, which, if you remember, was the first phase that we looked at, and the whole process can restart. So that's basically how mitosis works. So if we were to do it again, basically this one would go over here and we'd focus on this one. They would duplicate. Then the spindle fibers would come in. They would separate them. Cytokinesis would split the cells apart. And again, we'd have two brand new cells, which this is how cells replicate and replicate and replicate, giving you tissues throughout the body.